More than 350 prescription medications intended to treat diseases have been found to induce liver injury as a side effect. This liver injury can fall under hepatic cytolysis, steatosis, and cholestasis. These effects, if caused by medication, is broadly termed DILI, or drug-induced liver injury. This can be so serious it requires a liver transplant for the patient or otherwise lead to death. Although it is unclear the exact mechanism of DILI, it always seems to be linked with mitochondrial dysfunction caused by the drug. In this video, we'll talk about two main mechanisms of mitochondrial dysfunction and focus on the effects of acetaminophen as an example. Mitochondria can oxidize fatty acids and other substances. This is especially important in the liver, which oxidizes and detoxifies substances in the bloodstream. This is called MTFAO, or beta oxidation. 4 sequential reactions split one acetyl-CoA molecule from the fatty acid, which becomes shortened. These reactions are repeated and repeated until there is only a ketone body left that gets oxidized via the TCA cycle. MTFAO is a major source of ATP. Impairment of these reactions lead to energy shortage, which then leads to hepatocyte necrosis, or death of the liver cells, as well as cytolytic hepatitis. Because the fatty acids are not being oxidized, it also leads to hepatic steatosis, the accumulation of lipids in the liver. Since lower acetyl-CoA levels reduce the activity of pyruvate carboxylase, which is important for gluconeogenesis, it can also cause hypoglycemia. Inhibition of the MRC, otherwise known as the ETC, also causes significant ATP shortage, which then leads to hepatocyte necrosis. Since the ETC is dysfunctional, no FADH2 or NADH is oxidized, so FAD and NAD+ are not being regenerated, causing secondary impairment of MTFAO and the TCA cycle. Acetaminophen, aka paracetamol, aka APAP, is usually considered safe. A small amount of APAP is oxidized by cytochrome P450 in mitochondria to NAPK. NAPK is usually detoxified by hepatic GSH, but when you overdose APAP, GSH stores cannot keep up, and free NAPK covalently binds to mitochondrial proteins, leading to ETC impairment, which then causes all the terrible effects described earlier. How do we know this? Acyl-carnitine derivatives from incomplete MTFAO are found after overdose of APAP.